Okay, thanks for the organizer for having me here. And I'm maybe one of the examples of disruptive science. I'm going to talk about some studies that we are doing about perception of quantum light. This is kind of a misleading name because all light can be described by quantum mechanics, right? But you're going to understand that in a bit. Okay, so the idea of this project is that it mixtures quantum mechanics and neuroscience, two of the main fields of science nowadays, right? Quantum mechanics on one side describes very well the microscopic world, not only that, but it do it very well. It generates a lot of technologies so far, right? The laser, the old transistor-based technologies, the cell phone, the computers, right? So those are the first generations of technologies. And on the other side, we have neuroscience, the studies of the nervous system, the brains, the spiral cord, the disorder, the functions, right? So we are trying to work in the intersection between them. What is the intersection? We are trying to discover that, right? So let's see. So the idea of this project is exactly that, to close the gap between these two fields. And for that, we will use quantum optics tools, is what I know, quantum optics. I'm just starting in the field of biophysics, so I work mostly on that side. So the idea is to work with quantum optics tools to generate different states of light. I know how to do that. Then we will send that light to visual perception of human beings, okay? And the idea is to study conscious and unconscious human visual perception. And for that, we will use perceptual and cognitive neuroscience. Okay, so this is our disruptive project. Okay, the team is this one. On the quantum mechanics side, we have people from Federal University of Rio de Janeiro and Federal University of Minas Gerais. Okay. Gabriela Barreto Lemos is leading this project, so I decided to put her in green there. And on the neuroscience side, people from IDOR and University of São Paulo, and there are some external collaborators too. So my research, I work, as I mentioned earlier, I work with quantum optics, so I work with light prepared in a process called spontaneous parametric Dunn conversion. So the idea is that you send a laser, maybe it's more precise, this guy here. So send a laser to a nonlinear crystal, and then some of the photons of that laser are annihilated, and two photons are created in modes A and B. Okay, so you annihilate some photons, create other two, but that should happen with some natural limitations, right? Like conservation of energy, conservation of moment. And that makes, those natural limitations makes that these photons to have some correlations, okay? To have some entanglement depending on the preparation. I won't go into detail of that, but the idea is that they are correlated. And these two photons are born together at the same time, okay? Okay, so the idea is that in my lab, we have very good control on different degrees of freedom of light, polarization. We can manipulate the polarization using wave plates, Pockels cells. Also, the transfer spatial degrees of freedom. We can, for example, convert this pattern into this pattern of the light of a laser beam, for example. We can convert that using this kind of modulators, spatial light modulators. We can also control the time, but using these phase modulator, amplitude modulators, also these Pockels cells. And also the frequency. You can convert the frequency of the photons from one frequency to the other using these phase modulators. Obviously, all these things are done with some efficiency, okay? So there's some loss there in those processes. And all this control is so far used for quantum information, to build a quantum computer, to build better sensors, more precise sensors, or to build quantum communication systems, quantum cryptographic systems, such that you guarantee inviolable communications, and obviously to attack some problems such as the Schrödinger cat or the Bell inequalities, etc. So far, these photons that I produce are being studied in this context, and what we are proposing here is to study them in other contexts related with these questions, okay? So this is exactly the intersection between quantum optics and neuroscience. So the idea is two of the main questions of this project are what is our sensitivity in detecting photons? You know, the idea is that, I will explain that in a few slides, but other question, are our eyes sensitive to different types of light statistics? So those are like questions that we are, and inside those questions we have other questions that could be interesting, how the sensitivity is altered for different regions of retina. Are there spatial temporal correlation in photon detection? There are other questions that are interesting, and we are trying to answer in this project. Okay, and on this side, does this depend on the region of retina, the light statistics? How do we study this, right? Like, those are questions that, and we started this project 3 years ago, and we already have a lab that Sergio showed in his slides. And we are, I think, I trust that, I think we can have some result in a few months, maybe 1 year. But for the first question, it's not a new question at all. Hedge and his collaborators in 1932, they published that paper when they already they mentioned that that our eyes can detect between 5 and 7 photons. Okay, so it's not a new question. And then was a long time without so many publications. And then in 2016 came this paper here claiming that we can detect single photons. And that was like kind of, wow, it's just a nature communication. Why, right? Like it would be like a huge result. But then there were some other groups. This is a renowned scientist in quantum optics, and he published this correspondence here saying, however, we argue that this work suffers from 3 major issues: self-contradicting results, inappropriate statistical analysis, and critical lack of statistical power. So they are talking about this paper here. Okay, so this question is not answered at all. Okay, so it's something that we are going to research in the following years. And what about photon statistics? I will describe what I mean with that. It's maybe not clear for you all. So the idea is that we have different sources of light, right? The thermal bulbs are one of the possible light sources, the laser, and then we have the single photon emitters, the atoms, the quantum dots, the NV centers. Okay, there are many examples. And the idea that depending on the source, you have different kind of statistics. For example, the laser emits the light in a Poissonian distribution. So when you go and study what happened with the distribution with the variance of the photons and compare it with the mean number, you find that they are equal because they respect this kind of statistics. Okay. Then this kind of bulbs, right? They respect this kind of of distribution is like a Bose-Einstein distribution. And for this kind of distribution, what you have is that the variance is larger than the mean photon number, okay? And for sub-Poissonian, it happened the opposite. The variance is smaller than the mean photon number. So these are three kinds of statistics, and the idea is to shine our eyes with this different kind of statistic and see what happened, okay? And how do you know that those statistics, those lights have different statistics? You use one kind of function that are called the autocorrelation functions. Were proposed by Roe Glauber in 1963, if I'm not wrong, and he obtained the Nobel Prize in 2005 because of this contribution. Okay, so the idea is that he developed a formalism to characterize different types of light, different statistics of light. Okay. So the idea, how do you measure the statistic? The idea is that you send the light you want to study to a beam splitter, okay, a common beam splitter, the one that we put in a Michelson interferometer, right? And you divide that light into into two outputs of the beam splitter, and you detect that light there with two detectors, okay? And you count the single counts that detects basically the amount of photons reaching that detector, and also the coincidences, the events where you have one photon detected and one detector and one photon in the other detector. That is an event called coincidence, okay? And basically with this simple setup, you can compute this function, and with the value of this function, you can decide what kind of statistics that light has, okay? When you go to the low regime, you can basically compute this function by obtaining the probability of a coincidences between 1 and 2, and then the probability of detecting one photon in 1 at time t1, and one photon in 2 at time t2. Okay, so we're going to go in this direction, like in the low photon regime. Okay. So now let's see what happened with these light statistics in the temporal regime, right? So we have the time in this direction, and these black circles are photons. So the idea is that this super Poissonian distribution, the one of the thermal bulbs, right? What you have is instantaneous intensities, like all the photons are bunched, are like they like to be together, okay? Sometimes you have events with less photons, but what you have, the characteristics of this super Poissonian light is instantaneous high intensities, okay? Then the Poissonian distribution, the laser, right? You have a like random distribution. Sometimes they are bunched, sometimes they are not bunched, so you have something that is random. And for the case of sub-Poissonian, you have this distribution. Let's say that you pump a single photon emitter with a pulse laser, and what you have is an emission of one photon each time you receive a pump. Okay, so you have this distribution that is completely organized, right? Okay. So the idea then is that you have this correlation function bigger than 1 for the bunching, G2, the correlation function equal to 1 for just coherent light for the laser, and then this G2 smaller than 1 for the single photon emitters. Okay, so the idea is that we will send these three types of light, actually these two and another one that I will talk in a bit, to the eyes and see what is the response. Okay, so the third light that we are planning to send is this light that I know very well, that is the spontaneous parametric light. Okay, so the idea is that we generate, each time that we send a pulse, we generate 2 photons, and then since this source is not efficient, you are sending this repetition rate, right? And sometimes you create a pair, okay? But you know that every time you have light here, you have a pair of photons. So it's what we call a super bunch light. All the time you have a pair, okay? So this is the other light that we will send. And the idea is that the G2, in this case, increase a lot. It's very large. It's above 500. For example, in this paper, we showed something larger than this. And for the case of the thermal light, the case of the sun or the light bulbs, is around 2, the G2, okay? So, so this is the idea of the project, right? So to send, on one hand, the spontaneous parametric down conversion light, then to send laser, and then send light bulb. And the idea is that we have to create, right, those light, one of the main goals, right, is to create that light indistinguishable in the rest of the degrees of freedom. The only thing that should vary from one light to the other is just the statistics. And that is challenging, and we are working on that now, okay? So my presentation was, the idea was not presenting results, was to present a new line of research and new ideas, and I really would like to discuss with you if you have some proposals, some new ideas related with this project. I'm open to discuss, okay? So, but just to mention that we are not crazy. This was already investigated in this biologic system. Actually, is in this paper they took the eyes of this Xenopus levis frog, and they sent different kind of statistics, different light statistics to the eyes, and they measure this psychometric curve. And with this psychometric curve, given that you have different slope here, you can conclude that this guy actually is sensitive to the statistics of light. So this is like a preliminary result. We are trying to go a little bit farther doing this in the human eye, okay? So this is what I have to tell you. Our financial support is Pioneer Science. I didn't translate it, but thanks so much.
Okay, questions?
I have one comment and one question.
Yeah.
There is a professor, John Cameron, he's already deceased, and he was a pioneer in medical physics. And he was surprised that our body can sense almost anything except ionized radiation. So one time he decided to do an experiment in himself. He put an X-ray source in front of the eye, did all the calculations about radio protection because he was an expert on that, and looked to the radiation source and could not see anything. But I don't incentivize anybody to do that, right? I'm just telling, you know, how important it is this. Yeah, don't try at home. And the other question is, from what I understand, I mean, you are just wondering if your neurological system can distinguish in between different statistics of stimuli.
Yeah.
And this could be applied not only for light, but for other direct stimuli. Is that correct? I mean, I don't know about sound, because sound seems to be very time-locked. There is a specialist there. I don't know other kinds of sound.
Like, yeah, vibrations, right? Like
the question
vibrations.
I'm asking this is because I'm involved with a project that we call Neuromat. Mm-hmm. And the idea is to study how the brain works. And there is this hypothesis that we somehow have in our brain statistical structures that once we see a stimuli that somehow matches that memory we have, we react very fast. So that's why we have some reaction times that if you do all the physical principles calculation, it won't be possible to do that. But if a car is coming, you know, a Puma is coming, he knows how to rush away, right, from the puma because, you know, you have this imprinted. So can you also expand your research to these other areas?
Yeah, so far we are restricted to that, but I think that all these new questions are welcome for sure. Yeah, I think that we are focused to answer these questions, but yeah, we can for sure extend this research to other kind of detection. And the idea behind this is that maybe why we cannot create biologic single photon detectors. We know that, for example, superconducting systems are good for detecting single photon, right? But why? We don't know if biological systems are also, right? So this is maybe one of the possible applications, right, behind the. Okay.
Very nice proposal. But I have not, maybe I have not seen that, but what kind of biological model you use in your project?
Okay, yes.
You say
yes.
human eyes, but I am
yes,
not sure.
So the idea is to send the light to human beings. And that's why we have a neuroscience team, right, and a psychophysics team, and this is integrated. This will be with them. Obviously, we will maybe participate because we know about the quantum mechanics size and what we are sending and what kind of stimuli we are sending, right? But is this multidisciplinary field.
Your apparatus for the human.
We don't have a model so far. Yeah, we have to see some results to see how we model it, right?
So, thank you very much. Do you know if the people that did the frog paper have considered the human case? Because it seems like a natural next step.
I think that in that case was different because they isolated a single rod photosensor, so it was a simplified visual system. Okay, so I think that maybe we can start doing something like that. I don't know, like it's open. Okay, but in that case, it's simpler. You have just a rod cell, and you measure the photocurrent there. Okay, so it's.
Before I get there, I have a quick question. Is
Yeah.
there a biological relevance to detecting the statistics of the photons? Biological relevance? I don't know. I mean, it's.
Look, I think, as answering your question, I think that we are prepared to detect thermal light like this, light from the sun, that is the bunched light. We are prepared for that. We are all the time in contact with that light. But we don't know, right, what happened with the other kind of lights. So, yeah, it's answering that question. Yeah.
Hi. I don't know if you would know how to answer this question because it's quite biological, but do you know how the photoreceptors that we have in our eyes, if they have like the time that they have to wait until changing conformation to detect another photon? Do you think that these like the cells that perceive the light, they have to integrate the signal of multiple photoreceptors? Do you think this talks to your project as well?
So you have the rods and the cones, right? We are working with cones. The idea, if you go to single photon detector and sensitivity of our eyes, then are rod cells, okay? But you are talking about the dark time of the detector. I think the dark time, you have it in all the detectors. You have that. You need time to put the system available for the detection, right? So that happened in the superconducting and the semiconducting detector and also in the biological. Okay.
Okay, thank you again, Gabriel.
Thank you.
We have coffee break now.